Hello, my name is Indra Castillo, and I am in cell biology. This PowerPoint is also for cell biology, and we are doing this PowerPoint on the spectroscopic ruler, fluorescence resonance energy transfer, which is also known as FRET. So, I know what you're wondering. What is fluorescence resonance energy transfer, aka FRET? So, to understand it, we should go back to the beginning. Fluorescence resonance energy transfer was a theoretical analysis developed by Theodor Forster, and it became a physical phenomenon over 50 years ago. It relies on distance-dependent transfer of energy from a donor molecule to an acceptor molecule. Due to its sensitivity to nature, it's been used to investigate molecular interactions and is the radiationless transmission of energy from a donor molecule to an acceptor molecule. But how does it work? So we're going to be referring to the picture on this screen to understand how it works. So a donor group acts like a chromophore, aka a dye, and is excited by a photon, and then it relaxes to the lowest excited singlet state, S1. If the acceptor group is not too far, the energy released when the electron returns to the ground state, S0, may simultaneously excite the donor group, and this is known as resonance. After excitation, the excited acceptor emits a photon and returns to the ground state. This transfer of energy leads to a reduction in the donor's fluorescence intensity and excited state lifetime, and an increase in the acceptor's emission intensity results in a pair of molecules that interact in such a manner that FRET occurs, and it's often referred to as a donor-acceptor pair. So I just said a lot of words, but I'm gonna, shorten it. Pretty much, basically, energy created by fluorescence excitation of one molecule is transferred to an adjacent molecule. And if the two molecules are photophores, the excitation from the first molecule, the donor, results in fluorescence emission of the second molecule, the acceptor. But there are requirements for FRET. The molecules must be less than 10 nanometers apart, and the donor emissions must overlap for acceptor excitation to even show up. So, where is FRET used? It's used to measure the dynamic activities of biological molecules within a nanoscale. Because of this, the technique is used and applied in many fields, including single molecular experiments, molecular motors, biosensors, and DNA mechanical movements. The reason they're used in these fields and applications is because FRET tells us about molecular interactions, ligand binding to a receptor, protein-protein interactions, and the most important, which is also used in cancer often, molecular dimerization. So, in the research paper titled Non-Invasive In Vivo Imaging of Near-Infrared Label Transferrin in Breast Cancer Cells and Tumors Using Fluorescence Lifetime FRET, the overall question in the research was, can a combined technique of both FRET and near-infrared fluorescence lifetime technique be capable of non-invasively detecting internalized forms of transferrin in cancer cells or tumors in a small live animal model? So for this, you can see in the screen a picture, and that is actually one of the figures from this research paper. So how did they design this figure, or experimentally design for this figure? They quantified bound and internalized transferrin utilizing an intensity-based quantitative confocal microscopy FRET in vitro assay based on the binding of acceptor and donor label transferrin to the homodimeric transferrin R at the plasma membrane by using standard methods. They also used laser microscopy to focus the intensity of these interactions, in which transferrin complexes were concentrated using concentrators that excluded flow-through particles larger than 35 kD. So if you look at the figure, you see that the transferrin and transferrin R FRET assays show similar behavior in both the T47D breast cancer cells and the HMEX. By the way, the HMEX stands for the human mammary epithelial cells, and you can see it on the first row. The T47D has all the crazy blue lights, and the HMEX has only a few lights in the first two parts. But you can see in part A pretty much that the T47D breast cancer cell have a lot more uptake in both the acceptor and the quenched donor section, which is the first two parts. So what this pretty much is telling us, and overall, is that the pseudo-color images of acceptors, quenched donor, The uncorrected fret, aka U fret, and corrected fret, position fret, or known as P fret on the screen, shows the typical endocytic pattern of irregular and puncture structures. So in the P fret images, it confirms the existence of transfers between the indigenous donor and acceptor in intracellular puncture in the endocytic structures. So I just said a lot of crazy stuff, but I'm going to pretty much tell you what this means. Pretty much, according to all of his data, including A, B, and C, by the way, B and C simplifies everything in quantitative modes, talking about ratios. But overall, in this data, it means that the acceptor and quench donor images in both the T47 cells and the HMX cells show transfer and uptake. However, it does show that the T47 cells show significantly higher levels of transfer internalization versus the control HMX, which was in the part A. So both of them were actually organized, if you can see in the picture, like they're kind of clustered all together. So this showed clustered pattern. And this was expected due to the hemodimeric nature of transferrin R. And they also exhibited cluster behavior in the receptor ligand complexes through the endocytemic trafficking pathways, which was also expected. However, despite the higher expression level of transferrin R in the T47D cells, these interactions actually remained organized in clusters. And the weirdest part was that they didn't show any significant increase in the fret levels. In the fret levels overall. So apparently, according to these results, I'm going to say overall, they demonstrated the difference between the transferrin R expression levels between cancer cells and versus the normal cells. And it's reflected in the detection of fret levels, which can be seen in the higher intensity levels of acceptor labeled transferrin. This also established that the intensity-based transferrin R to transferrin fret assay can be used to quantitatively distinguish cells expressing higher levels of transferrin R found in cancer cells from non-cancerous cells that show lower levels of transferrin R expression. Basically, they were able to discriminate cancer cells from normal cells by thresholding fret at higher acceptor intensity levels. So here's a good question. What's the basic strengths and drawbacks of the experiment? So one of the successes overall from the experiment was they were able to discriminate cancer cells, just like I said, from normal cells by thresholding fret at higher acceptor intensity levels. However, a drawback from this experiment is that it's not clear why that even though there was a higher expression level in transferrin R in T47D cells, there was no significant increase in fret levels between organized clusters, and they did, vary in size. So a question I had after reading this research was that the technique, it's pretty much great to understand the interactions between two molecules, or maybe, in the research paper, like they said, the interactions of a certain molecule or ligand to a drug, or in this case, that was transferrin. But a really good question that I had, or I guess a pondering, if you will, was I was wondering if there was any research on how they could expand the field, like to better understand the interactions between a complex or a community, or kind of like, when the cells are quorum sensing for certain bacterias. I wonder if they could do that, like if they could just, make it a bit where they could not see just cell to cell, but rather like a small group of cell to cells to see how they better interact with one another, not just as individuals. So that'd be a good question, I think. Below are the following references I used. The first three are pretty much just to, they help me understand what fret was, and the last reference is just the reference to the research paper. And that is all, I guess. Thank you for listening.